Hello, my name is Mark PEAKMAN. I trained as a clinical immunologist, spent many years as a clinical academic in the area of immunology. And I'm going to talk today about the future and challenges in immune inflammation. Currently, I'm head of autoimmunity and type 1 diabetes in the cluster in the therapeutic area of immunology and inflammation with Sanofi, based in Cambridge, Massachusetts. So on the face of it, it looks like we have great success at the moment in therapeutics for autoimmunity and inflammatory disease. If you look on the left graph here, showing the global immunology market, at the moment we're around $100 billion of revenue for pharma companies for therapeutics that are focused on autoimmune and inflammatory diseases. And that's set to rise to perhaps 150 billion in a short space of time, and it seems to be roughly doubling over 7 or 8 years. However, if we look a little bit deeper into this, and these are mainly biologics and protein conjugates. If we look a little bit deeper, we can see that actually this revenue is based on around 4 or 5 major diseases: rheumatoid arthritis, psoriasis, inflammatory bowel disease, and ankylosing spondylitis. And within those diseases, the typical response rate for a patient is around 30%. So first of all, we have an unmet need in the patients where we have success, in that we only have a small proportion that make a clinical response. Secondly, we have a large unmet need because we're only looking at a small proportion of those diseases that are autoimmune in nature and could be tractable with these therapies. So it's estimated by the American Autoimmune Disease Association that there are over 100 diseases that come into this classification of autoimmunity, such that around 1 in 14 people in the US has an autoimmune inflammatory disease. That's over 23 million people. And yet these 4 or 5 top indications only represent less than 50% of these diseases. So there's another large unmet need there. We're not reaching the populations that have the disease burden. So how can we address that? In 10 minutes, of course, I can't give you all the answers. I'm going to give you my perspective on this particular question. So I'm highlighting some of the challenges here and then trying to think about what the solutions are. And I'm going to focus just on 3 particular areas that I'm interested in my own work. The first is, can we make sure we get the right drug to the right patient at the right time? So it's all about focus and it's all about precision. Secondly, can we shift this focus on these large revenue yielding diseases that are the top 5 and move into the unmet need in other autoimmune diseases? And I think that's all about getting the risk and benefit right. Clearly, when you have severe autoimmune diseases, you are prepared to take the risk of a drug that is essentially immune suppressive. And there might be other diseases where you're not prepared to take such a risk. So can we work on the risk benefit so that we can actually benefit a greater proportion of these diseases? And then finally, can we address the challenges through new biology? And I'll give one example there, again, that interests me from my own background, a focus on restoring immune homeostasis. So to start with focus and precision, I want to highlight some of the exciting opportunities that there are currently, and in particular, the area of public-private partnerships to address these problems. So these are large issues. They're important to academics, they're important to patients, they're important to healthcare providers, and they're important to pharma. But they're too big for any one of those units to tackle alone. So these public-private partnerships are a very important way of addressing these big areas of understanding biology and understanding the pharmacology. And this particular public-private partnership, called 3TR, focuses on responder, non-responder biology in the field of autoimmune and inflammatory diseases and immunotherapeutics. It's called 3TR because it's studying the taxonomy, in other words, classification of diseases. It's looking at how we can optimize therapeutics, and it's looking at the identification of novel targets that might be the focus of therapeutics in the future. and really trying to understand how we can get patients into permanent remission. It's a public-private partnership. It's the largest in this organization called IMI, the Innovative Medicines Initiative in the European Union, over 80 million euros. It's got a very large number of centers in Europe across 15 countries. And I'm really delighted that the scientific project lead for this sits in my cluster in Cambridge, Massachusetts, Terry Means, who's doing a fantastic job of coordinating this particular effort. So we look at 7 different diseases. And if you look at the mission statement, you'll see fundamentally what the goal is here. It's to increase knowledge around the molecular pathways and mechanisms that are linked to response and non-response, that 30% that is key in different immune-mediated allergic and inflammatory diseases. And of course, focusing most of our efforts on the 70% where we don't see a clinical response. And this is tapping into an area that has become really quite interesting and important for therapeutics. The idea of precision medicine actually here now being played out as precision immunotherapy. And it's based on our understanding over the last years that human disease is very heterogeneous. And what that boils down to is the concept of disease endotypes. So these are subtypes of disease that we can define by having distinct functional or pathobiological mechanisms. And importantly, I think in the definition, we need to include the fact that that endotype is tractable by a particular therapeutic. So how does this play out in a large consortium like 3TR? Well, you might collect data on large numbers of patients across different diseases, looking at multiple immune phenotypes. And I give an example here. One phenotype might be responsiveness to drug X. One might be measurement of immune regulation or autoantibodies, the role of interferons, CD4 T cells, CD8 T cells, innate immune activation, and so on. And you would essentially grade each subject in each of these areas using a sort of signal strength measure. And from that, if you apply, you have large enough numbers of individuals in a disease or diseases, you apply essentially a kind of principal component analysis. The expectation is that patients will start to cluster where their disease biology is leading them. And for example, here, patients with a predominant type 1 interferon signature would cluster separately, for example, from those with predominant CD4 T cell activation. And both of those would need completely different therapeutic approaches. For example, through emerging biologics that blockade type 1 interferons, and the very well-established approach to co-stimulation blockade. And of course, it would now make absolute sense to take these diseases, understand where a patient sits in this matrix, and give them a particular type of therapy. And then you will see that your 30% response rate is much higher. And that is really the understanding that is at the heart of 3TR. Moving on to my second sort of area of challenge and solution, how do we get a better handle on risk and benefit? If you think about what we're doing with our top 5 target diseases that I mentioned earlier, you know, most of that revenue that I showed in the graph in the last 10 or 15 years has been around a very small number of biologics. For example, anti-TNF or B-cell depletion through anti-CD20. And what we know about those is they're very effective on blocking immune function, but they are systemic, and that's where the off-target effects come and where the unwanted side effects come. And that's why these are not being deployed in some of the other diseases. So the question is whether we could get smarter therapeutics where we exploit the warhead here of anti-TNF or anti-CD20 or whatever it is, but we place it in the tissue that we're actually interested in controlling the immune response in. And that would avoid the off-target effects. And so I wanted to pick up an example that's been in the news recently. It's a therapeutic being developed by a company called Pandion. And actually, in the time that I was preparing these slides, Pandion was bought by Merck, which shows you that I think this is an emerging area of great interest. It's enormously facilitated, of course, by the engineering that we can now undertake to manufacture bispecifics, trispecifics, multispecifics, and so on. So this particular example from Pandion is a PD-1 agonist as its warhead, so it will switch off activated T cells. And On the other hand, on the other side of the molecule, through a scaffold, is a moiety that will bind to MADCAM, the mucosal addressing cell adhesion molecule. And on the right-hand side here, I've captured some of Pandion's data, showing that if you inject this therapeutic in vivo and then take tissues and perform ex vivo analysis, you can see the presence of the MADCAM moiety binding to lymph nodes and vessels in the target tissues, shown in red. You can see co-localization then of the PD-1 agonist moiety in green, and you merge these on the right-hand side, and you can see that in the tissues now we have our therapeutic. It is not systemic, it's available locally. And I think this is going to be an important emerging area of getting these powerful immunosuppressants into the right tissues. And finally, I wanted to just touch upon another strategy for the future, certainly emerging now, and that is, can we look at restoring immune homeostasis in a different way? So the majority of the current drugs, their MOA, their mechanism of action, is to suppress or deplete immune effector pathways. And of course, what every immunologist knows is that there is another side to the effector pathway coin, and that is immune regulation. So the biology that's emerged in the last 10, 15 years has given us an enormous understanding of the many facets of immune regulation, but in particular, one cell type stands out, and we understand a lot more about this now, the regulatory T cell, or Treg. And Tregs depend upon a particular cytokine, interleukin-2, for their function. And so a number of companies, including Sanofi, have started to look at this area very seriously. And Sanofi has a unique biosynthetic platform where we're using a new genetic code to manufacture molecules. These are called synthorins. And through this, we have manufactured a novel synthetic interleukin-2 that, because of the positioning of the PEG molecule, does not bind to the IL-2 receptor beta chain, but binds to the IL-2 receptor alpha chain, and therefore provides very high Treg selectivity, as you'll see in the cartoon in the middle. The effector T cells don't bear the IL-2 receptor alpha chain, whereas the Tregs do, and therefore they are the ones that get the benefit of this cytokine when it's administered. So as you see in the top right graph, in a preclinical model, when we administer this molecule in vivo, we get really very impressive and robust expansion of regulatory T cells into the peripheral blood. These are entirely bona fide Tregs by any measurement that we can give, including functional measurements. And then you can see the bottom right, if we administer these into an antigen-specific inflammatory model, represented here by immunization with keyhole limpid hemocyanin, and then challenge with that molecule in the ear tissue, you can see that we can control inflammation in a dose-dependent fashion with a very small frequency of injections of our synthetic IL-2 molecule. So I think this is going to be an important area. A number of companies are positioning themselves with these IL-2 based therapies to really tackle the homeostasis that we want to restore from a different angle than depletion or modification of effector T cells. So to summarize, we have shown, I think, that there is fantastic promise to transform the lives of patients who have immune and inflammatory diseases, but there is a lot of work still to do. And I think we're going to build on that success by smarter use of the current strategies with an increasing emphasis on precision immunotherapy. And there are multiple new technologies that are continuing to excite the industry, and I've just given you one example of those. Thank you for your attention, and very happy to take questions.
Thank you very much, Mark PEAKMAN, for this very exciting presentation, where we can now have a better idea of all the challenges you have with this approach and these approaches and these strategies. Well, the precision immunotherapy approach makes a lot of sense. We could hear that, getting the right drug for the right patient and so on. So what does it take to implement such a strategy? Is it simple?
It's anything but simple, Fabrice. It's a great question. So I think I highlighted, that if we're going to do this, we need to form very large partnerships. The reason for that is that we need large numbers of patients to make sense of the data. We need fantastic access to all of the what we call metadata around those patients. We need access to biological samples to perform the analyses. Patients have to be really well, what we call curated, very, very careful physician approaches to the selection of the patients. We need to do that and then perform, these sophisticated analyses. And quite often we will do it once and we will get some exciting data, and then we'll need to do it again for validation. So it takes time. And then when you have identified the subjects that you think might be amenable to a particular therapy, there's no way to go forward except to test that out. So you have to go into the clinic and do that. You will probably do that without selecting them out initially, but you'd have that as one of your major objectives of the study, and then capture that. And then, of course, if you're successful, which is fantastic, you need to operationalize that. So you need to talk to the regulatory authorities if you're a pharma company. You need to develop a co-diagnostic or an assay that will allow you to stratify patients in the way that you want. And then you need to get that out to the market. So I can see this stretching out for a number of years. I think that what we need to, the prize we need to keep our eye on here is that we're addressing this non-responsive group. That I'm less worried about 30% where we get a response. It's the 70% where we don't. So I think this is the real challenge here, is to find other aspects of the biology of a disease, and to I'm sure we have the therapeutics available. It's just having then the opportunity to put them into play. So I think it's a long journey.
Yeah, we can understand it, and to address such challenges and to reach that group, you mentioned the promise of new technologies. Are there any other things on the horizon that you're excited about?
Yeah, I'm very excited about an area that is just beginning to emerge. That I think puts us in a new place for therapies. So we have molecules that deactivate, we have or block, we have molecules that capture, we have molecules that deplete cells or molecules, but there's still what we call a number of undruggable targets or difficult to drug targets for various reasons. Sometimes because they sit in complex families where we can't get the precision of the targeting. And so there are a couple of new strategies essentially that work around degrading those target molecules. It's called PROTAC, and what you do is have a sort of approach where you can target a molecule, and then you're almost putting a tag on it that is telling the cell to recycle it and get it degraded. And I think these are going to be a new addition to the armamentarium for immune therapeutics. So very excited to see that biology emerging.
All these exciting emerging technologies or strategies costs a lot. It's expensive, and you show that expenditure on drugs in this space is going up and up. And is that really sustainable?
So I think that's another challenging question. I think we have to tackle that from two sides. One is, and it's something that I know pharma looks at very carefully, is cost of goods. Can we make therapeutics smarter, cheaper? Of course, if we use them in the right patients, that's also going to make them, essentially, you're using less drug. And then I think that we have to be aware of the patient burden. And I've looked at this in a number of diseases. It goes beyond some of the things we think about. It affects development, education, their role in society, jobs, their productivity. So there are lots of unmeasurable aspects to a chronic illness. And I think when we factor those in, I think we'd look at the cost-benefit ratio in a completely different way. So I'm not despondent about us hitting a ceiling. I think these are very worthwhile therapies. We just have to make sure they're safe and effective.
Thank you very much, Mark PEAKMAN, for all these exciting things that you're working on. We are cross finger now. We hope that it will success and succeed, and that you get the money and everything to go on. Thanks a lot.
Thank you.